Biology's most exciting era. Welcome to biology, the scientific study of life. You are becoming involved with biology during its most exciting era. The largest and best-equipped community of scientists in history is beginning to solve biological puzzles that once seemed unsolvable. We are moving ever closer to understanding how a single microscopic cell develops into a complex plant or animal, how plants convert solar energy to the chemical energy of food, how the human mind works, how various forms of life network in biological communities such as forests and coral reefs, and how the great diversity of life on Earth evolved from the first microbes. The more we learn about life, the more fascinating it becomes, as progress on one question leads to even more questions that will captivate curious minds for decades to come. More than anything else, biology is a quest, an ongoing inquiry about the nature of life. Modern biology is as important as it is inspiring. Research breakthroughs in genetics and cell biology are transforming medicine and agriculture. Molecular biology is providing new tools for fields as diverse as anthropology and criminal science. Neuroscience and evolutionary biology are reshaping psychology and sociology. New models in ecology are helping societies evaluate environmental issues, such as the causes and biological consequences of global warming. These are just a few examples of how biology weaves into the fabric of our culture more than ever before. There has never been a better time to explore life. The phenomenon we call life defies a simple one-sentence definition. Yet almost any child perceives that a dog or a bug or a plant, such as the fern fiddlehead, that graces the cover of this book is alive, while a rock is not. We recognize life by what living things do. As we set off to explore life, it helps to have a panoramic view of the vast field of study before us. This opening chapter introduces the wide scope of biology, highlights the diversity of life, describes themes such as evolution that unify all of biology, and examines methods of inquiry that biologists use to explore life. Biologists explore life from the microscopic to the global scale. The study of life extends from the microscopic scale of the molecules and cells that make up organisms to the global scale of the entire living planet. We can divide this enormous range into different levels of biological organization. A hierarchy of biological organization. Imagine zooming in from space to take a closer and closer look at life on Earth. Our destination is a forest in Ontario, Canada, where we will eventually use microscopes and other instruments to explore a maple leaf right down to the molecular level. The biosphere. As soon as we are near enough to Earth to make out its continents and oceans, we begin to see signs of life, in the green mosaic of the planet's forests, for example. This is our first view of the biosphere, which consists of all the environments on Earth that are inhabited by life. The biosphere includes most regions of land, most bodies of water, such as oceans, lakes, and rivers, and the atmosphere to an altitude of several kilometers. Ecosystems. As we approach Earth's surface for an imaginary landing in Ontario, we can begin to make out a forest with an abundance of deciduous trees, trees that lose their leaves in one season and grow new ones in another. Such a deciduous forest is an example of an ecosystem. Grasslands, deserts, and the ocean's coral reefs are other types of ecosystems. An ecosystem consists of all the living things in a particular area, along with all the non-living components of the environment with which life interacts, such as soil, water, atmospheric gases, and light. All of Earth's ecosystems combined make up the biosphere. The entire array of organisms inhabiting a particular ecosystem is called a biological community. The community in our forest ecosystem includes many kinds of trees and other plants, a diversity of animals, various mushrooms and other fungi, and enormous numbers of diverse microorganisms, which are living forms such as bacteria that are too small to see without a microscope. Each of these forms of life is called a species. A population consists of all the individuals of a species living within the bounds of a specified area. For example, our Ontario forest includes a population of sugar maple trees and a population of American black bears. We can now refine our definition of a community as the set of populations that inhabit a particular area. Organisms. Individual living things are called organisms. Each of the maple trees and other plants in the forest is an organism, and so is each forest animal, such as a frog, squirrel, bear, and insect. The soil teems with microorganisms such as bacteria, organs, and organ systems. The structural hierarchy of life continues to unfold as we explore the architecture of the more complex organisms. A maple leaf is an example of an organ, a body part consisting of two or more tissues, which we'll see upon our next scale change. Stems and roots are the other major organs of plants. Examples of human organs are the brain, heart, and kidney. The organs of humans and other complex animals are organized into organ systems, each a team of organs that cooperate in a specific function. For example, the human digestive system includes such organs as the tongue, stomach, and intestines. Tissues. Our next scale change to see a leaf's tissues requires a microscope. The leaf on the left has been cut on an angle. The honeycombed tissue in the interior of the leaf, upper half of photo, is the main location of photosynthesis, the process that converts light energy to the chemical energy of sugar and other food. We are viewing the sliced leaf from a perspective that also enables us to see the jigsaw puzzle-like tissue called epidermis, the skin on the surface of the leaf, lower half of photo. The pores through the epidermis allow the gas carbon dioxide, a raw material for sugar production, to reach the photosynthetic tissue in the interior of the leaf. At this scale, we can also see that each tissue has a cellular structure. In fact, each kind of tissue is a group of similar cells. Cells. The cell is life's fundamental unit of structure and function. Some organisms, such as amoebas and most bacteria, are single cells. Other organisms, including plants and animals, are multicellular. Instead of a single cell performing all the functions of life, a multicellular organism has a division of labor among specialized cells. A human body consists of trillions of microscopic cells of many different kinds, including muscle cells and nerve cells, which are organized into the various specialized tissues. For example, muscle tissue consists of bundles of muscle cells. And note again the cells of the tissue within a leaf's interior. Each of the cells you see is only about 25 micrometers across. It would take more than 700 of these cells to reach across a penny. As small as these cells are, you can see that each contains numerous green structures called chloroplasts, which are responsible for photosynthesis. Organelles. Chloroplasts are examples of organelles, the various functional components that make up cells. In this figure, a very powerful tool called an electron microscope brings a single chloroplast into sharp focus. Molecules. Our last scale change vaults us into a chloroplast for a view of life at the molecular level. A molecule is a chemical structure consisting of two or more small chemical units called atoms, which are represented as balls in this computer graphic of a chlorophyll molecule. Chlorophyll is the pigment molecule that makes a maple leaf green. One of the most important molecules on Earth, chlorophyll absorbs sunlight during the first step of photosynthesis. Within each chloroplast, millions of chlorophylls and other molecules are organized into the equipment that converts light energy to the chemical energy of food. A closer look at ecosystems. Life does not exist in a vacuum. Each organism interacts continuously with its environment, which includes other organisms as well as non-living factors. The roots of a tree, for example, absorb water and minerals from the soil. The leaves take in carbon dioxide from the air. Solar energy absorbed by chlorophyll drives photosynthesis, which converts water and carbon dioxide to sugar and oxygen. The tree releases oxygen to the air, and its roots help form soil by breaking up rocks. Both organism and environment are affected by the interactions between them. The tree also interacts with other life, including soil microorganisms associated with its roots and animals that eat its leaves and fruit. Ecosystem dynamics. The dynamics of any ecosystem include two major processes. One process is the cycling of nutrients. For example, minerals acquired by plants will eventually be returned to the soil by microorganisms that decompose leaf litter, dead roots, and other organic debris. The second major process in an ecosystem is the flow of energy from sunlight to producers to consumers. Producers are plants and other photosynthetic organisms that convert light energy to chemical energy. Consumers are organisms, such as animals, that feed on producers and other consumers. Energy conversion. Moving, growing, reproducing, and other activities of life require organisms to perform work. And work depends on a source of energy. The exchange of energy between an organism and its surroundings often involves the transformation of one form of energy to another. For example, when a leaf produces sugar, it converts solar energy to chemical energy in sugar molecules. When an animal's muscle cells use sugar as fuel to power movements, They convert chemical energy to kinetic energy, the energy of motion. And in all these energy conversions, some of the available energy is converted to thermal energy, which working organisms dissipate to their surroundings as heat. In contrast to chemical nutrients, which recycle within an ecosystem, energy flows through an ecosystem, usually entering as light and exiting as heat. A closer look at cells. In life's structural hierarchy, the cell has a special place as the lowest level of organization that can perform all activities required for life. For example, the ability of cells to divide to form new cells is the basis for all reproduction and for the growth and repair of multicellular organisms. Your every movement and thought are based on the activities of muscle cells and nerve cells. Even a global process such as the recycling of carbon, a chemical element essential to life, is the cumulative product of cellular activities, including the photosynthesis that occurs in the chloroplasts of leaf cells. Understanding how cells work is a major research focus of modern biology. The cell's heritable information. Take another look at the dividing cell. Within the cells, you can see structures called chromosomes, which are stained with a blue glowing dye. The chromosomes are partly made of a substance called deoxyribonucleic acid, or DNA for short. DNA is the substance of genes, the units of inheritance that transmit information from parents to offspring. Your blood group, AB, or O, for example, is the result of certain genes that you inherited from your parents. Each chromosome has one very long DNA molecule, with hundreds or thousands of genes arranged along its length. The DNA of chromosomes replicates as a cell prepares to divide. Thus, each of the two cellular offspring inherits a complete set of genes. Each of us began life as a single cell stocked with DNA inherited from our parents. Replication of that DNA transmitted those genes to our trillions of cells. In each cell, the genes along the length of DNA molecules encode the information for building the cell's other molecules. In this way, DNA directs the development and maintenance of the entire organism. The molecular structure of DNA accounts for its information-rich nature. Each DNA molecule is made up of two long chains arranged into what is called a double helix. Each link of a chain is one of four kinds of chemical building blocks called nucleotides. The way DNA encodes a cell's information is analogous to the way we arrange the letters of the alphabet into precise sequences with specific meanings. The word rat, for example, conjures up an image of a rodent. The words tar and art, which contain the same letters, mean very different things. Libraries are filled with books containing information encoded in varying sequences of only 26 letters. We can think of nucleotides as the alphabet of inheritance. Specific sequential arrangements of these four chemical letters encode the precise information in genes, which are typically hundreds of thousands of nucleotides long. One gene in a bacterial cell may be translated as build a purple pigment. A particular human gene may mean make the hormone insulin. More generally, most genes program the cell's production of large molecules called proteins. The sequence of nucleotides along each gene codes for a specific protein with a unique shape and function in the cell. One protein might be part of the contractile apparatus of muscle cells. Another protein might be an antibody, part of the body's defense system against viruses and other disease agents. Still another protein might be an enzyme, a protein that catalyzes, speeds up, a specific chemical reaction in a cell. Almost all cellular activities involve the action of one or more proteins. DNA provides the heritable blueprints, but proteins are the tools that actually build and maintain the cell. All forms of life employ essentially the same genetic code. A particular sequence of nucleotides says the same thing to one organism as it does to another. Differences between organisms reflect differences between their nucleotide sequences. But because the genetic code is universal, it is possible to engineer cells to produce proteins normally found only in some other organism. One of the first pharmaceutical products obtained using this technology was human insulin, produced by bacteria into which a gene for this human protein was inserted. The entire library of genetic instructions that an organism inherits is called its genome. The chromosomes of each human cell pack a genome that is about 3 billion nucleotides long. If the one-letter symbols for this sequence of nucleotides were written in letters the size of those you are now reading, the genetic text would fill about 600 books the size of this one. Within this genomic library of nucleotide sequences, are genes coding for the production of more than 75,000 different kinds of proteins, each with a specific function. All cells share certain characteristics. For example, every cell is enclosed by a membrane that regulates the passage of materials between the cell and its surroundings, and every cell uses DNA as its genetic information. We can distinguish two main forms of cells: prokaryotic cells and eukaryotic cells. The cells of two groups of microorganisms called bacteria and archaea are prokaryotic. All other forms of life, including plants and animals, are composed of eukaryotic cells. A eukaryotic cell is subdivided by internal membranes into various membrane-enclosed organelles, including the chloroplasts. In most eukaryotic cells, the largest organelle is the nucleus, which contains the cell's DNA as chromosomal molecules. The other organelles are located in the cytoplasm, the entire region between the nucleus and outer membrane of the cell. Prokaryotic cells are much simpler and generally smaller than eukaryotic cells. In a prokaryotic cell, the DNA is not separated from the rest of the cell by enclosure in a membrane-bounded nucleus. Prokaryotic cells also lack the other kinds of membrane-enclosed organelles that characterize eukaryotic cells. The prokaryote-eukaryote difference is an example of the biological diversity we will explore. But first, let's take one more look at the hierarchy of biological order, this time in the context of a research movement called systems biology. Biological systems are much more than the sum of their parts. The whole is greater than the sum of its parts. That familiar adage tours the important concept that a combination of components can form a more complex organization called a system. Examples of biological systems are cells, organisms, and ecosystems. To understand how such systems work, it is not enough to have a parts list, even a complete one. The future of biology is in understanding the behavior of whole integrated systems. The emergent properties of systems. Take another look at the levels of life. With each step upward in this hierarchy of biological order, novel properties emerge that are not present at the level just below. These emergent properties are due to the arrangement and interactions of parts as complexity increases. For example, a test tube mixture of chlorophyll and all the other molecules found in a chloroplast cannot perform photosynthesis. The process of photosynthesis emerges from the very specific way in which the chlorophyll and other molecules are arranged in an intact chloroplast. To take another example, if a serious head injury disrupts the intricate architecture of a human brain, the mind may cease to function properly even though all of the brain parts are still present. Our thoughts and memories are emergent properties of a complex network of nerve cells. At an even higher level of biological organization, at the ecosystem level, the recycling of nutrients, such as carbon, depends on a network of diverse organisms interacting with each other and with the soil and air. Emergent properties are neither supernatural nor unique to life. We can see the importance of arrangement in the distinction between a box of bicycle parts and a working bicycle. And while graphite and diamonds are both pure carbon, they have very different properties based on how their carbon atoms are arranged. But compared to such non-living examples, the emergent properties of life are particularly challenging to study because of the unrivaled complexity of biological systems, the power and limitations of reductionism. Because the properties of life emerge from complex organization, scientists seeking to understand biological systems confront a dilemma. On the one hand, we cannot fully explain a higher level of order by breaking it down into its parts. A dissected animal no longer functions. A cell reduced to its chemical ingredients is no longer a cell. Disrupting a living system interferes with the meaningful understanding of its processes. On the other hand, something as complex as an organism or a cell cannot be analyzed without taking it apart. Reductionism, reducing complex systems to simpler components that are more manageable to study, is a powerful strategy in biology. For example, by studying the molecular structure of DNA that had been extracted from cells, James Watson and Francis Crick inferred in 1953 how this molecule could serve as the chemical basis of inheritance. In 2001, almost half a century after the famous work of Watson and Crick, an international team of scientists published a rough draft of the sequence of 3 billion chemical letters in a human genome. Researchers have also sequenced the genomes of many other species. The press and world leaders acclaimed the Human Genome Project as one of the greatest scientific achievements ever. But unlike past cultural zeniths, such as the moonwalk of Apollo astronauts, the sequencing of the human genome is more a commencement than a climax. As the quest continues, scientists are learning the functions of thousands of genes and their protein products. And research is now moving on to how the activities of these myriad molecules are coordinated in the development and maintenance of cells and whole organisms. At the cutting edge of this research is the approach called systems biology. Systems biology. Biology is turning in an exciting new direction as many researchers begin to complement reductionism with new strategies for understanding the emergent properties of life. How all the parts of biological systems, such as cells, are functionally integrated. This changing perspective is analogous to moving from ground level on a certain street corner to an aerial view above a city, where you can now see how variables such as time of day, construction projects, accidents, and traffic signal malfunctions affect traffic dynamics throughout the city. The ultimate goal of systems biology is to model the dynamic behavior of whole biological systems. Accurate models will enable biologists to predict how a change in one or more variables will impact other components and the whole system. How, for example, will a slight increase in a muscle cell's calcium concentration affect the activities of the dozens of proteins that regulate muscle contraction? How will a drug that lowers blood pressure affect the function of organs throughout the human body and possibly cause harmful side effects? Systems biology is relevant to the study of life at all levels. Scientists investigating ecosystems pioneered the systems approach in the 1960s with elaborate models diagramming the network of interactions between species and non-living components in salt marshes and other ecosystems. Even earlier, biologists studying the physiology, functioning of humans and other organisms were integrating data on how multiple organs coordinate processes such as the regulation of sugar concentration in the blood. Systems biology is now taking hold in the study of life at the cellular and molecular levels, driven partly by the deluge of data from the sequencing of genomes and the growing catalog of known protein functions. In 2003, for example, a large research team published a network of protein interactions within the cell of a fruit fly, a popular research organism. The model is based on an extensive database of thousands of known proteins and their known interactions with other proteins. For example, protein A may bind to and alter the activities of proteins B, C, and D, which then go on to interact with still other proteins. The basics of the system strategy are straightforward enough. First, it is necessary to inventory as many parts as possible, such as all the known genes and proteins in a cell, reductionism. Then it is necessary to investigate how each part behaves in relation to others in the working system, all the protein-protein interactions, in the case of our fly cell example. Though the basic idea of systems biology is simple, the practice is not, as you would expect from the complexity of biological systems. It has taken 3 key research developments to bring systems biology within reach. High-throughput technology. Systems biology depends on methods that can analyze biological materials very rapidly and produce enormous volumes of data. Such mega data collection methods are said to be high throughput. Bioinformatics. The huge databases that result from high throughput methods would be chaotic without the computing power, software, and mathematical models to process and integrate all this biological information. The new field of bioinformatics is extracting useful biological information from the enormous, ever-expanding datasets, such as DNA sequences and lists of protein interactions. Interdisciplinary research teams. In 2003, Harvard Medical School formed a Department of Systems Biology, its first new department in 2 decades. Nearby MIT is busy organizing over 80 faculty members from many departments into a new program for computational and systems biology. These and other systems biology startups are melting pots of diverse specialists, including engineers, medical scientists, chemists, physicists, mathematicians, computer scientists, and biologists from a variety of fields. A number of prominent scientists are promoting systems biology with missionary zeal, but so far the excitement exceeds the achievements. However, as systems biology gathers momentum, it is certain to have a growing impact on the questions biologists ask and the research they design. After all, scientists aspired to reach beyond reductionism, to grasp how whole biological systems work long before new technology made modern systems biology possible. In fact, Decades ago, biologists had already identified some of the key mechanisms that regulate the behavior of complex systems such as cells, organisms, and ecosystems. Feedback regulation in biological systems. A kind of supply and demand economy applies to some of the dynamics of biological systems. For example, when your muscle cells require more energy during exercise, they increase their consumption of the sugar molecules that provide fuel. In contrast, when you rest, a different set of chemical reactions converts surplus sugar to substances that store the fuel. Like most of the cell's chemical processes, those that decompose or store sugar are accelerated or catalyzed by the specialized proteins called enzymes. Each type of enzyme catalyzes a specific chemical reaction. In many cases, these reactions are linked into chemical pathways, each step with its own enzyme. How does the cell coordinate its various chemical pathways? In our specific example of sugar management, how does the cell match fuel supply to demand by regulating its opposing pathways of sugar consumption and storage? The key is the ability of many biological processes to self-regulate by a mechanism called feedback. In feedback regulation, the output or product of a process regulates that very process. In life, the most common form of regulation is negative feedback, in which accumulation of an end product of a process slows that process. For example, the cell's breakdown of sugar generates chemical energy in the form of a substance called ATP. An excess accumulation of ATP feeds back and inhibits an enzyme near the beginning of the pathway. Though less common than negative feedback, there are also many biological processes regulated by positive feedback, in which an end product speeds up its production. The clotting of your blood in response to injury is an example. When a blood vessel is damaged, structures in the blood called platelets begin to aggregate at the site. Positive feedback occurs as chemicals released by the platelets attract more platelets. The platelet pile then initiates a complex process that seals the wound with a clot. Feedback is a regulatory motif common to life at all levels, from the molecular level to the biosphere. Such regulation is an example of the integration that makes living systems much greater than the sum of their parts. 1. Emergent properties and sentence construction. The relationship between a sentence and the alphabet of letters from which it is constructed is a classic example of emergent properties. The individual letters of the alphabet have no inherent meaning on their own, but when combined in a specific way, they form words, phrases, and sentences that convey complex ideas and meanings. The meaning of a sentence is an emergent property that arises from the arrangement and interaction of its constituent parts, letters and words. Just as the properties of life emerge from the complex organization of biological systems, the meaning of a sentence emerges from the organization of letters and words. High-throughput technology and bioinformatics. High-throughput technology and bioinformatics are complementary because they work together to analyze and interpret large amounts of biological data. High-throughput technology generates vast amounts of data quickly and efficiently, while bioinformatics provides the computational tools and methods to analyze, integrate, and interpret this data. Bioinformatics helps to extract meaningful insights and patterns from the data, which can then be used to inform further research and experimentation. In other words, high-throughput technology provides the data, and bioinformatics provides the tools to make sense of it. 3. Regulatory mechanism in a toilet. The regulatory mechanism at work in a toilet is an example of negative feedback. When the water level in the tank reaches a certain height, the float rises and triggers the lever to shut off the water valve, preventing the tank from overflowing. This is similar to negative feedback in biological systems, where an excess of a product or substance inhibits a process or pathway. In both cases, the feedback mechanism helps to maintain homeostasis and prevent excessive accumulation of a substance, water in the toilet tank or a product in a biological pathway. Biologists explore life across its great diversity of species. We can think of biology's enormous scope as having two dimensions. The vertical dimension, which we examined in this chapter's first two concepts, is the size scale that reaches all the way from molecules to the biosphere. But biology's scope also has a horizontal dimension stretching across the great diversity of species, now and over life's long history. Diversity is a hallmark of life. Biologists have so far identified and named about 18 million species. This enormous diversity of life includes approximately 5,200 known species of prokaryotes. 100,000 fungi, 290,000 plants, 52,000 vertebrates, animals with backbones, and 1,000 insects, more than half of all known forms of life. Researchers identify thousands of additional species each year. Estimates of the total species count range from about 10 million to over 200 million. Whatever the actual number, the vast variety of life makes biology's scope very wide. Grouping species, the basic idea. There seems to be a human tendency to group diverse items according to similarities. For instance, perhaps you organize your music collection according to artist. And then maybe you group the various artists into broader categories such as dance music, party music, exercise music, and study time music. In the same way, grouping species that are similar is natural for us. We may speak of squirrels and butterflies, though we recognize that many different species belong to each group. We may even sort groups into broader categories such as rodents, which include squirrels, and insects, which include butterflies. Taxonomy, the branch of biology that names and classifies species, formalizes this ordering of species into a series of groups of increasing breadth. You will learn more about this taxonomic scheme in Chapter 25. For now, we will focus on kingdoms and domains, the broadest units of classification. The three domains of life. Until the last decade, most biologists adopted a taxonomic scheme that divided the diversity of life into 5 kingdoms, including the plant and animal kingdoms. But new methods, such as comparing the DNA sequences of diverse species, have led to an ongoing reevaluation of the number and boundaries of kingdoms. Different researchers have proposed anywhere from 6 kingdoms to dozens of kingdoms. But as debate continues at the kingdom level, there is more of a consensus that the kingdoms of life can now be grouped into 3 even higher levels of classification called domains. The 3 domains are named bacteria, archaea, and eukarya. The first 2 domains, domain bacteria and domain archaea, both consist of prokaryotes, organisms with prokaryotic cells. Most prokaryotes are unicellular and microscopic. All the eukaryotes, organisms with eukaryotic cells, are now grouped into the various kingdoms of domain eukarya. In the era of the 5-kingdom scheme, most of the single-celled eukaryotes, including the microorganisms known as protozoans, were placed in a single kingdom, the kingdom Protista. The recent taxonomic trend has been to split the protists into several kingdoms. In addition to these protistine kingdoms, the domain eukarya includes 3 kingdoms of multicellular eukaryotes, the kingdoms Plantae, Fungi, and Animalia. These 3 kingdoms are distinguished partly by their modes of nutrition. Plants produce their own sugars and other foods by photosynthesis. Fungi are mostly decomposers that absorb nutrients by breaking down dead organisms and organic wastes, such as leaf litter and animal feces. Animals obtain food by ingestion, which is the eating and digesting of other organisms. Unity in the diversity of life. As diverse as life is, there is also evidence of remarkable unity, especially at the molecular and cellular levels. An example is the universal genetic language of DNA, which is common to organisms as different as bacteria and animals. And among eukaryotes, unity is evident in many features of cell structure. How can we account for life's dual nature of unity and diversity? The process of evolution, introduced in the next concept, illuminates both the similarities and differences among Earth's life. 1. How is a mailing address analogous to biology's hierarchical taxonomic system? A mailing address is analogous to biology's hierarchical taxonomic system, in that both involve a hierarchical structure with increasingly specific categories. Just as a mailing address includes a country, state, city, and street address, the taxonomic system includes domain, kingdom, phylum, class, order, family, genus, and species. 2. What is the key difference that distinguishes organisms of domain eukarya from the other 2 domains? The key difference that distinguishes organisms of domain eukarya from the other 2 domains is the presence of eukaryotic cells, which have a true nucleus and other membrane-bound organelles. In contrast, the domains bacteria and archaea consist of prokaryotic cells, which lack a true nucleus and other membrane-bound organelles. Evolution accounts for life's unity and diversity. The history of life, as documented by fossils and other evidence, is a saga of a changing Earth billions of years old, inhabited by an evolving cast of living forms, This evolutionary view of life came into sharp focus in November 1859, when Charles Robert Darwin published one of the most important and controversial books ever written, entitled On the Origin of Species. By natural selection, Darwin's book was an immediate bestseller and soon made Darwinism almost synonymous with the concept of evolution. The origin of species articulated two main points. First, Darwin presented evidence to support his view that contemporary species arose from a succession of ancestors. Darwin called this evolutionary history of species descent with modification. It was an insightful phrase, as it captured the duality of life's unity and diversity, unity in the kinship among species that descended from common ancestors, diversity in the modifications that evolved as species branched from their common ancestors. Darwin's second main point was to propose a mechanism for descent with modification. He called this evolutionary mechanism natural selection. Darwin synthesized his theory of natural selection from observations that by themselves were neither new nor profound. Others had the pieces of the puzzle, but Darwin saw how they fit together. He inferred natural selection by connecting two readily observable features of life. Darwin called this mechanism of evolutionary adaptation natural selection, because the natural environment selects for the propagation of certain traits. Darwin's theory of natural selection summarizes the ability of natural selection to edit a population's heritable variations. Take another look at the skeletal architecture of the bat's wings. These forelimbs, though adapted for flight, actually have all the same bones, joints, nerves, and blood vessels found in other limbs as diverse as the human arm, the horse's foreleg, and the whale's flipper. Indeed, all mammalian forelimbs are anatomical variations of a common architecture, much as the flowers are variations on an underlying orchid theme. Such examples of kinship connect life's unity in diversity to the Darwinian concept of descent with modification. In this view, the unity of mammalian limb anatomy reflects inheritance of that structure from a common ancestor, the prototype, mammal from which all other mammals descended. Their diverse forelimbs modified by natural selection operating over millions of generations in different environmental contexts. Fossils and other evidence corroborate anatomical unity in supporting this view of mammalian descent from a common ancestor. Thus, Darwin proposed that natural selection, by its cumulative effects over vast spans of time, could enable an ancestral species to split into two or more descendant species. This would occur, for example, if one population fragmented into several subpopulations isolated in different environments. In these various arenas of natural selection, one species could gradually radiate into many species as the geographically isolated populations adapted over many generations to different sets of environmental factors. The family tree of 14 finches illustrates a famous example of adaptive radiation of new species from a common ancestor. Darwin collected specimens of these birds during his 1835 visit to the remote Galapagos Islands, 900 kilometers (km) off the Pacific coast of South America. These relatively young volcanic islands are home to many species of plants and animals found nowhere else in the world, though Galapagos organisms are clearly related to species on the South American mainland. After volcanism built the Galapagos several million years ago, finches probably diversified on the various islands from an ancestral finch species that by chance reached the archipelago from the mainland. Years after Darwin's collection of Galapagos finches, researchers began to sort out the relationships among the finch species, first from anatomical and geographic data, and more recently with the help of DNA sequence comparisons. Biologists' diagrams of evolutionary relationships generally take tree-like forms, and for good reason. Just as an individual has a genealogy that can be diagrammed as a family tree, each species is one twig of a branching tree of life extending back in time through ancestral species more and more remote. Species that are very similar, such as the Galapagos finches, share a common ancestor at a relatively recent branch point on the tree of life. But through an ancestor that lived much farther back in time, finches are related to sparrows, hawks, penguins, and all other birds. And birds, mammals, and all other vertebrates, animals with backbones, share a common ancestor even more ancient. We find evidence of still broader relationships in such similarities as the matching machinery of all eukaryotic cilia. Trace life back far enough, and there are only fossils of the primeval prokaryotes that inhabited Earth over 35 billion years ago. We can recognize their vestiges in our own cells, in the universal genetic code, for example. All of life is connected through its long evolutionary history. Editing is a better metaphor than creating, because natural selection doesn't create new traits or variations. Instead, it acts on existing variation in a population, favoring individuals with traits that enhance their survival and reproduction. This process edits out individuals with traits that are less beneficial, leading to an increase in the frequency of favorable traits in the population over time. The three domains you learned about in Concept 13 can be represented in the tree of life as the three main branches. On the eukaryotic branch, three of the sub-branches are the kingdoms Plantae, Fungi, and Animalia. Evidence supports the view that fungi and animals are more closely related to each other than either of these kingdoms is to plants. Draw a simple branching pattern that symbolizes the relationship between these three eukaryotic kingdoms. Eukarya. Eplanti. At fungi and Animalia. This branching pattern shows that fungi and Animalia share a more recent common ancestor with each other than with Plantae. Biologists use various forms of inquiry to explore life. The word science is derived from a Latin verb meaning to know. Science is a way of knowing. It developed out of our curiosity about ourselves, other life forms, the world, and the universe. Striving to understand seems to be one of our basic urges. At the heart of science is inquiry, a search for information and explanation, often focusing on specific questions. Inquiry drove Darwin to seek answers in nature for how species adapt to their environments. Inquiry is driving the analysis of genomes, which is helping us understand biological unity and diversity at the molecular level. In fact, the inquisitive mind is the engine that drives all progress in biology. There is no formula for successful scientific inquiry, no single scientific method with a rulebook that researchers must rigidly follow. As in all quests, science includes elements of challenge, adventure, and surprise, along with careful planning, reasoning, creativity, cooperation, competition, patience, and the persistence to overcome setbacks. Such diverse elements of inquiry make science far less structured than most people realize. That said, it is possible to distill certain characteristics that help to distinguish science from other ways of describing and explaining nature. Biology blends two main processes of scientific inquiry: discovery science and hypothesis-based science. Discovery science is mostly about describing nature. Hypothesis-based science is mostly about explaining nature. Most scientific inquiries combine these two research approaches. Sometimes called descriptive science, discovery science describes natural structures and processes as accurately as possible through careful observation and analysis of data. For example, discovery science gradually built our understanding of cell structure, and it is discovery science that is expanding our databases of genomes of diverse species. Observation is the use of the senses to gather information, either directly or indirectly with the help of tools such as microscopes that extend our senses. Recorded observations are called data. Put another way, data are items of information on which scientific inquiry is based. The term data implies numbers to many people, but some data are qualitative, often in the form of recorded descriptions rather than numerical measurements. Along with these qualitative data, researchers also collect quantitative data, which are generally recorded as measurements. Discovery science can lead to important conclusions based on a type of logic called induction, or inductive reasoning. Through induction, we derive generalizations based on a large number of specific observations. The sun always rises in the east is an example, and so is all organisms are made of cells. That generalization, part of the so-called cell theory, was based on 2 centuries of biologists discovering cells in the diverse biological specimens they observed with microscopes. The observations and inductions of discovery science engage inquisitive minds to seek natural causes and explanations for those observations. What caused the diversification of finches on the Galapagos Islands? What causes the roots of a plant seedling to grow downward and the leaf-bearing shoot to grow upward? What explains the generalization that the sun always rises in the east? In science, such inquiry usually involves the proposing and testing of hypothetical explanations, or hypotheses. The role of hypotheses in inquiry. In science, a hypothesis is a tentative answer to a well-framed question, an explanation on trial. It is usually an educated postulate, based on experience and the available data of discovery science. A scientific hypothesis makes predictions that can be tested by recording additional observations or by designing experiments. We all use hypotheses in solving everyday problems, let's say. For example, if your flashlight fails during a campout, that's an observation. The question is obvious. Why doesn't the flashlight work? Two reasonable hypotheses based on experience are that 1. The batteries in the flashlight are dead, or 2. The bulb is burnt out. Each of these alternative hypotheses makes predictions you can test with experiments. For example, the dead battery hypothesis predicts that replacing the batteries will fix the problem. Diagrams. This campground inquiry. Of course, we rarely dissect our thought processes this way when we are solving a problem using hypotheses, predictions, and experiments. But hypothesis-based science clearly has its origins in the human tendency to figure things out by working. Deduction. The if-then logic of hypothesis-based science. A type of logic called deduction is built into hypothesis-based science. Deduction contrasts with induction, which, remember, is reasoning from a set of specific observations to reach a general conclusion. In deductive reasoning, the logic flows in the opposite direction, from the general to the specific. From general premises, we extrapolate to the specific results we should expect if the premises are true. If all organisms are made of cells, premise 1, and humans are organisms, premise 2, then humans are composed of cells, deductive prediction about a specific case. In hypothesis-based science, deduction usually takes the form of predictions about what outcomes of experiments or observations we should expect if a particular hypothesis, premise, is correct. We then test the hypothesis by performing the experiment to see whether or not the results are as predicted. This deductive testing takes the form of if-then logic. In the case of the flashlight example, if the dead battery hypothesis is correct and you replace the batteries with new ones, then the flashlight should work. A closer look at hypotheses in scientific inquiry. The flashlight example illustrates two important qualities of scientific hypotheses. First, a hypothesis must be testable. There must be some way to check the validity of the idea. Second, a hypothesis must be falsifiable. There must be some observation or experiment that could reveal if such an idea is actually not true. The hypothesis that dead batteries are the sole cause of the broken flashlight could be falsified by replacing the old batteries with new ones. But try to devise a test to falsify the hypothesis that invisible campground ghosts are fooling with your flashlight. Does restoring flashlight function by replacing the bulb falsify the ghost hypothesis? Not if the playful ghosts are continuing their mischief. The flashlight inquiry illustrates another key point about hypothesis-based science. The ideal is to frame two or more alternative hypotheses and design experiments to falsify those candidate explanations. In addition to the two explanations tested in Figure 125, one of the many additional hypotheses is that both the batteries and the bulb are bad. What does this hypothesis predict about the outcome of the experiments in Figure 125? What additional experiment would you design to test this hypothesis of multiple malfunction? We can mine the flashlight scenario for still one more important lesson about hypothesis-based science. Although the burnt-out bulb hypothesis stands up as the most likely explanation, notice that the testing supports that hypothesis not by proving that it is correct, but by not eliminating it through falsification. Perhaps the bulb was simply loose and the new bulb was inserted correctly. We could attempt to falsify the burnt-out bulb hypothesis by trying another experiment, removing the bulb and carefully reinstalling it. But no amount of experimental testing can prove a hypothesis beyond a shadow of doubt because it is impossible to exhaust the testing of all alternative hypotheses. A hypothesis gains credibility by surviving various attempts to falsify it while testing eliminates, falsifies, alternative hypotheses. The myth of the scientific method. The steps in the flashlight example trace an idealized process of inquiry called the scientific method. We can recognize the elements of this process in most research articles published by scientists, but rarely in such structured form. Very few scientific inquiries adhere rigidly to the sequence of steps prescribed by the textbook scientific method. For example, a scientist may start to design an experiment, but then backtrack upon realizing that more observations are necessary in other cases. Puzzling observations simply don't prompt well-defined questions until other research projects place those observations in a new context. For example, Darwin collected specimens of the Galapagos finches, but it wasn't until years later, as the idea of natural selection began to gel, that biologists started asking key questions about their history. Moreover, scientists sometimes redirect their research when they realize they have been barking up the wrong tree by asking the wrong question. For example, in the early 20th century, much research on schizophrenia and manic depressive disorder, now called bipolar disorder, got sidetracked by focusing too much on how life experiences cause these maladies. Research on causes and potential treatments became more productive when refocused on how chemical imbalances in the brain contribute to mental illness. To be fair, such twists and turns in scientific inquiry become more evident with the advantage of historical perspective. A case study in scientific inquiry, investigating mimicry in snake populations. Now that we have highlighted the key features of discovery science and hypothesis-based science, you should be able to recognize these forms of inquiry in a case study of actual scientific research. The story begins with a set of observations and generalizations from discovery science. Many poisonous animals are brightly colored, often with distinctive patterns that stand out against the background. This is called warning coloration, because it apparently signals dangerous species to potential predators. But there are also mimics. These imposters look like poisonous species, but are actually relatively harmless. An example is the flower fly, a non-stinging insect that mimics the appearance of a stinging honeybee. What is the function of such mimicry? What advantage does it confer on the mimics? In 1862, British scientist Henry Bates proposed the reasonable hypothesis that mimics such as flower flies benefit when predators confuse them with the harmful species. In other words, the deception may be an evolutionary adaptation that evolved by reducing the mimic's risk of being eaten. The team investigated a case of mimicry among snakes that live in North and South Carolina. A poisonous snake called the eastern coral snake has warning coloration, bold, alternating rings of red, yellow, and black. Predators rarely attack these snakes. It is unlikely that predators learn this avoidance behavior, as a first strike by a coral snake is usually deadly. Natural selection may have increased the frequency of predators that have inherited an instinctive recognition and avoidance of the warning coloration of the coral snake. A non-poisonous snake named the scarlet king snake mimics the ringed coloration of the coral snake. Both king snakes and coral snakes live in the Carolinas, but the king snake's geographic range extends farther north and west into regions where no coral snakes are found. The geographic distribution of the Carolina snakes made it possible to test the key prediction of the mimicry hypothesis. Mimicry should help protect king snakes from predators, but only in regions where coral snakes also live. The mimicry hypothesis predicts that predators in non-coral snake areas will attack king snakes more frequently than will predators that live where coral snakes are present. Field experiments with artificial snakes. To test the mimicry hypothesis, researchers made hundreds of artificial snakes out of wire covered with a clay-like substance. They fashioned two versions of fake snakes, an experimental group with the red, black, and yellow ring pattern of king snakes, and a control group of plain brown artificial snakes as a basis of comparison. The researchers placed equal numbers of the two types of artificial snakes in field sites throughout North and South Carolina, including the region where coral snakes are absent. After 4 weeks, the scientists retrieved the fake snakes and recorded how many had been attacked by looking for bite or claw marks. The data fit the key prediction of the mimicry hypothesis. Compared to the brown artificial snakes, the ringed snakes were attacked by predators less frequently only in field sites within the geographic range of the poisonous coral snakes. Designing controlled experiments. The snake mimicry experiment provides an example of how scientists design experiments to test the effect of one variable by canceling out the effects of any unwanted variables, such as the number of predators in this case. The design is called a controlled experiment, where an experimental group, the artificial king snakes in this case, is compared with a control group, the brown artificial snakes. Ideally, the experimental and control groups differ only in the one factor the experiment is designed to test. In our example, the effect of the snake's coloration on the behavior of predators. What if the researchers had failed to control their experiment? Without the brown mock snakes as a control group, the number of attacks on the fake king snakes in different geographic regions would tell us nothing about the effect of snake coloration on predator behavior at the different field sites. Perhaps, for example, fewer predators attack the artificial king snakes in the eastern and southern field sites simply because fewer predators live there. Or maybe warmer temperatures in those regions make predators less hungry. The brown artificial snakes enabled the scientists to rule out such variables as predator density and temperature because those factors would have had equal effects on the control group and experimental group. Yet predators in the eastern and southern field sites attacked more brown artificial snakes than king snakes. The clever experimental design left coloration as the only factor that could account for the low predation rate on the artificial king snakes placed within the range of coral snakes. It was not the absolute number of attacks on the artificial king snakes that counted, but the difference between that number and the number of attacks on the brown snakes. A common misconception is that the term controlled experiment means that scientists control the experimental environment to keep everything constant except the one variable being tested. But that's impossible in field research and not realistic even in highly regulated laboratory environments. Researchers usually control unwanted variables not by eliminating them through environmental regulation, but by canceling their effects by using control groups. Limitations of science. Scientific inquiry is a powerful way to know nature, but there are limitations to the kinds of questions it can answer. These limits are set by science's requirements that hypotheses be testable and falsifiable, and that observations and experimental results be repeatable. Observations that can't be verified may be interesting or even entertaining, but they cannot count as evidence in scientific inquiry. The headlines of supermarket tabloids would have you believe that humans are occasionally born with the head of a dog, and that some of your classmates are extraterrestrials. The unconfirmed eyewitness accounts and the computer-rigged photos are amusing, but unconvincing. In science, evidence from observations and experiments is only convincing if it stands up to the criterion of repeatability. The scientists who investigated snake mimicry in the Carolinas obtained similar data when they repeated their experiments with different species of coral snakes and king snakes in Arizona. And you should be able to obtain similar results if you were to repeat the snake experiments. Ultimately, the limitations of science are imposed by its naturalism, its seeking of natural causes for natural phenomena. Science can neither support nor falsify hypotheses that angels, ghosts, or spirits, both benevolent and evil, cause storms, rainbows, illnesses, and cures. Such supernatural explanations are simply outside the bounds of science. Theories in science. It's just a theory. Our everyday use of the term theory often implies an untested speculation. But the term theory has a very different meaning in science. First, a scientific theory is much broader in scope than a hypothesis. This is a hypothesis. Mimicking poisonous snakes is an adaptation that protects non-poisonous snakes from predators. But this is a theory. Evolutionary adaptations evolve by natural selection. Darwin's theory of natural selection accounts for an enormous diversity of adaptations, including mimicry. Second, a theory is general enough to spin off many new specific hypotheses that can be tested. For example, Peter and Rosemary Grant of Princeton University were motivated by the theory of natural selection to test the specific hypothesis that the beaks of the Galapagos finches evolve in response to changes in the types of available food. And third, compared to any one hypothesis, a theory is generally supported by a much more massive body of evidence. Those theories that become widely adopted in science, such as the theory of natural selection, explain a great diversity of observations and are supported by an accumulation of evidence. In fact, scrutiny of general theories continues through testing of the specific, falsifiable hypotheses they spawn. In spite of the body of evidence supporting a widely accepted theory, scientists must sometimes modify or even reject theories when new research methods produce results that don't fit. For example, the five-kingdom theory of biology. Diversity began to erode when new methods for comparing cells and molecules made it possible to test some of the hypothetical relationships between organisms that were based on the theory. If there is truth, in science, it is conditional, based on the preponderance of available evidence. Model building in science. You may work with many models in your biology course this year. Perhaps you'll model cell division by using pipe cleaners or other objects as chromosomes. Or maybe you'll practice using mathematical models to predict the growth of a bacterial population. Scientists often construct models as less abstract representations of ideas, such as theories or natural phenomena, such as biological processes. Scientific models can take many forms, such as diagrams, graphs, three-dimensional objects, computer programs, or mathematical equations. The choice of a model type depends on how it will be used to help explain and communicate the object, idea, or process it represents. Some models are meant to be as lifelike as possible. Other models are more useful if they are symbolic schematics. For example, the simple diagram does a good job of modeling blood flow through the chambers of a human heart without looking anything like a real heart. A heart model designed to help train a physician to perform heart surgery would look very different. Whatever its design, the test of a model is how well it fits the available data, how comfortably it accommodates new observations, how accurately it predicts the outcomes of new experiments, and how effectively it clarifies and communicates the idea or process it represents. The culture of science. Movies and cartoons sometimes portray scientists as loners working in isolated labs. In reality, science is an intensely social activity. Most scientists work in teams, which often include both graduate and undergraduate students. And to succeed in science, it helps to be a good communicator. Research results have no impact until shared with a community of peers through seminars, publications, and websites. Both cooperation and competition characterize the scientific culture. Scientists working in the same research field often check one another's claims by attempting to confirm observations or repeat experiments. And when several scientists converge on the same research question, there is all the excitement of a race. Scientists enjoy the challenge of getting there first, with an important discovery or key experiment. The biology community is part of society at large, embedded in the cultural milieu of the times. For example, changing attitudes about career choices have increased the proportion of women in biology, which has in turn affected the emphasis in certain research fields. A few decades ago, biologists who studied the mating behavior of animals focused mostly on competition among males for access to females. More recent research, however, emphasizes the important role that females play in choosing mates. For example, in many bird species, females prefer the bright coloration that advertises a male's vigorous health, a behavior that enhances the female's probability of having healthy offspring. Some philosophers of science argue that scientists are so influenced by cultural and political values that science is no more objective than other ways of knowing nature. At the other extreme are people who speak of scientific theories as though they were natural laws instead of human interpretations of nature. The reality of science is probably somewhere in between, rarely perfectly objective but continuously vetted through the expectation that observations and experiments be repeatable and hypotheses be testable and falsifiable. Science, technology, and society. The relationship of science to society becomes clearer when we add technology to the picture. Though science and technology sometimes employ similar inquiry patterns, their basic goals differ. The goal of science is to understand natural phenomena. In contrast, technology generally applies scientific knowledge for some specific purpose. Biologists and other scientists often speak of discoveries, while engineers and other technologists more often speak of inventions. And the beneficiaries of those inventions include scientists, who put new technology to work in their research. The impact of information technology on systems biology is just one example. Thus, science and technology are interdependent. The potent combination of science and technology has dramatic effects on society. For example, discovery of the structure of DNA by Watson and Crick 50 years ago, and subsequent achievements in DNA science led to the many technologies of DNA engineering that are transforming a diversity of fields, including medicine, agriculture, and forensics. Perhaps Watson and Crick envisioned that their discovery would someday produce important applications, but it is unlikely that they could have predicted exactly what those applications would be. The directions that technology takes depend less on the curiosity that drives basic science than on the current needs and wants of people and on the social environment of the times. Debates about technology center more on, should we do it, than can we do it? With advances in technology come difficult choices. For example, under what circumstances is it acceptable to use DNA technology to check if people have genes for hereditary diseases? Should such tests always be voluntary, or are there any circumstances when genetic testing should be mandatory? Should insurance companies or employers have access to the information, as they do for many other types of personal health data? Such ethical issues have as much to do with politics, economics, and cultural values as with science and technology. But scientists and engineers have a responsibility to help educate politicians, bureaucrats, corporate leaders, and voters about how science works and about the potential benefits and risks of specific technologies. Induction and deduction are two types of reasoning. Induction involves making generalizations based on specific observations. Deduction, on the other hand, involves drawing specific conclusions from general premises. In other words, induction goes from specific to general, while deduction goes from general to specific. This statement is mostly true, but it could be improved. Scientists design controlled experiments to study the effect of a single variable by controlling for other variables that could affect the outcome. It's not possible to completely prevent all other factors from changing, but rather to minimize their impact on the experiment. The researchers placed some of their artificial snakes beyond the geographic range of coral snakes to test the hypothesis that mimicry would only be effective in areas where coral snakes are present. By comparing the results from areas with and without coral snakes, the researchers could determine if the mimicry was effective in reducing predation. A hypothesis is a specific, testable prediction or explanation for a phenomenon. A theory, on the other hand, is a well-supported and widely accepted explanation for a set of phenomena. A theory is not just a guess or a hunch, but rather a comprehensive framework that explains a large body of evidence. In science, theories are not opposed to facts, but rather are the frameworks that help to explain and organize those facts. A set of themes connects the concepts of biology. In some ways, biology is the most demanding of all sciences, partly because living systems are so complex and partly because biology is an interdisciplinary science that requires knowledge of chemistry, physics, and mathematics. Modern biology is the decathlon of natural science. And of all the sciences, biology is the most connected to the humanities and social sciences. As biology students, you are definitely in the right place at the right time. No matter what brings you to biology, you will find the study of life to be endlessly challenging and uplifting. But this ever-expanding subject can also be a bit intimidating, even to professional biologists. How, then, can beginning students develop a coherent view of life, instead of hopelessly trying to memorize the details of a subject that is now far too big to memorize? One approach is to fit the many things you learn into a set of themes that pervade all of biology. Ways of thinking about life that will still apply decades from now, when much of the specific information fossilized in any textbook will be obsolete. Write a sentence relating the theme of scientific inquiry to the theme of science, technology, and society. The theme of scientific inquiry is closely tied to the theme of science, technology, and society, as scientific discoveries and advances often lead to new technologies that can have significant impacts on society.